Okay, welcome everyone, and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. I'm Jiyang, and I am the host of this session. I'm also the chair of a computational biology department at St. Jude. It is my great pleasure today to welcome our speaker, Dr. Jiyang, who is a faculty member in our department. So let's go over a little bit housekeeping issues. First of all, everyone should be able to use the chat function to enter questions to all panelists, and we will collect these questions and answer them, at the end of the lecture. And Dr. Yu have kindly offered to stay on for the additional QA and after the 1 o'clock, and we will end at 1:30. So let me introduce our today's speaker, Dr. Yu. Dr. Jiyang Yu was received his bachelor's degree in computer science in Zhejiang University in China, and become a PhD, received his PhD in biomedical informatics at Columbia University under the tutorage of Dr. Andre Califano, who is really a pioneer of a systems biologist. He then joined Pfizer as a senior scientist and worked there for 4 years. So in 2016, I was able to convince Dr. Yu to rejoin the academic rank, and also convince him that systems biology can make a big impact in pediatric cancer. So he joined our department in 2016 and had really made a major impact through his innovative work on the hidden driver algorithm that forms systems biology based data integration to identify things that cannot be discovered by regular differential expression or comparison or even mutation analysis. So it was with great enthusiasm, I'd like to introduce Dr. Yu to present his talk so that we can all learn the mystery about hidden driver. Jiyang.
All right. Thank you so much, Jinghui, for your kind introduction. And I want to thank Charles and Charlie for this wonderful opportunity. So I'm truly honored to present in this prestigious, lecture series and share some of our work on identifying and targeting hidden drivers for cancer precision medicine, particularly for pediatric cancer. So let me start with a real patient story. Reported in Science, June 2020, Miss Kiptos, who unfortunately had metastatic HER2-positive breast cancer, exhausted all possible treatment options, including HER2 target agents and latest immunotherapy. However, nothing could stop her tumor growth and spreading until she was invited into a clinical trial, Masana, which used a drug named ruxolitinib, a JAK inhibitor approved to treat bone marrow diseases, never been used for breast cancer treatment. And she had a miracle response with almost no side effects. So 3 months later, Nature reported the same story, and the PI underlying the story was Dr. Andrea Califano. As Jinghui mentioned, I had the fortune to be trained with Andrea. He has been my lifelong mentor friend since then. And Kevin Kalinske was a molecular oncologist who ran the trial. And I'm proud to tell you that that was based on my part of my PhD work, in which we identify interleukin-6, JAK2, STAT3 as vulnerability in subset of breast cancer independent of the JAK mutation, in close collaboration with Hotel Silva, who is a breast cancer biologist. When I first, heard that story, of course, years, before the media report, it was one of the most rewarding moments in my career, especially as condition biologist. So your discovery could impact patient lives. And here are 2 other recent translation stories that came out of my own group. One is a gene collaboration cell where we identify HDAC6 might be a therapeutic target for subset breast cancer. We finished a phase 1 trial with manuscript in revision against independent HDAC6 mutation. A second story was in wonderful collaboration with Jiyang here in Jude, where we identified the setnib, ABL inhibitor approved to treat leukemia with ABL fusion, actually effective in more than 44% of pediatric TL T-cell lymphoblastic leukemia, which has no ABL fusion. And through targeting LCK in a genetic independent fashion as well. And multiple clinical trials have been launched, to be launched. So the common theme of this story is the three stories just mentioned is that the driver activity, the vulnerability, and also the response are not driven by genetic or genomic alteration, which actually dominate the current cancer precision medicine paradigm. Mutations identified as molecules developed and is given to patients with that mutation. Has remarkable success, ALK inhibitor, EGFR inhibitor, many others. However, there are few substantial limitations with this approach. First, actual mutation is kind of rare. If you look at the mapping rate of NCR match trial, less than 4% of patients can be matched to target therapy based on the mutation profile. Even the patient can be matched, the response rate is surprisingly low. You may be shocked by the response rate reporting in NCR in a match trial. And in many cancers, over 50% of patients, they don't have any known cancer mutations. And the big ones, oncology and tumor suppressors, if you make, they're still undruggable despite the exciting advancement in PROTAC molecular glue type therapeutics. And many patients, which does not have the mutation may respond very well. So all of this has motivated us and many others to develop, approaches going beyond genome, identify drivers, therapeutic targets that could benefit more patients. So today I'm going to show you one hidden driver system biology approach my group has been developing for this. The basic idea is that we integrate multi-omics data, specific RNA and bark and single cell level, to reverse engineer the molecular networks, different layer, intracellular transcription factor replication network, CD network, as well as intracellular gene cell-cell communication network enabled by single cell data, and try to dissect the network rewiring from healthy to disease, for example, and try to pinpoint the underlying hidden driver that can be translated into potential therapeutic clinical applications. And we have been applying this kind of framework to a variety of contexts, from the basic immunology, development biology to pathologic conditions, including cancer. And key concept here is a hidden driver. So this is what I meant by a hidden driver. There are a lot of genes, proteins that are not, genetic altered, no, or epigenetic altered, no different expression mRNA or even at total protein level, but they are functionally important for the phenotype through post-transition modification, other mechanisms. Many of those actually are druggable, druggable ones fall into this category. And if you look at the information chain on the left, from DNA to RNA to protein and down to the phenotype, it's not hard to understand why they exist. There are way many steps, along this kind of information chain. What we measure by, sequencing mostly, it's really tiny tip of iceberg for what's happening in the cell, in tissue, in organ, in system. So therefore, give rise to the existence of hidden driver that conventional technology approach may fail to identify. And the challenge really is how we can utilize existing omics data, for example, RNA-seq, bark, and single cell, which we worked a lot to expose such hidden drivers. So next I'm going to describe you algorithm we developed to infer hidden driver from bark, omics data, net-based. On one hand, we have been going broader, extending to single cell, given the power of single cell sequence data, and also on the other hand, going deeper, focus on specific hidden driver and translate them into potential clinical applications. I will show you two exciting stories and also our efforts on developing combination therapy along this line. So NetBait was co-developed with Dr. Homberg's immunology department at St. Jude. It really started with a simple immunology question posed by Homberg for a long time. Conventionally, two subsets of dendritic cell CD positive negatively function distinguishably. However, the underlying molecular mechanism is unclear. So Homberg did transcriptome, whole proteome, or even false proteome profiling, try to understand what's the difference between the two subsets. Unfortunately, the conventional different expression or even positive enrichment analysis could not find anything that move forward. So he came to me, and we developed the NetBait by adding an extra layer of network inferred from a cohort of dendritic cell specific gene expression profiles. Then we superimpose the dendritic cell specific here seeding network into different layers of different expression, for example. Then you can see a lot of overlap to the different layers. And while the top enriched pathway, actually HAPO pathway, almost half of the HAPO kinases show up among the top kinase drivers identified. It's probably showing too much detail, but just mention that if you look at evidence from the transcriptomics and histoproteomics, you don't see any pattern of STK4, MST1 high expression, high activity in the CD positive dendritic cells. However, the network inferred by NetBait from the dendritic cell specific, sequencing network can show the black-white difference. So it really shows the power to capture those proteins that conventional approach may fail to identify, even phosphoproteomics here. And we have been promoting this kind of idea in the emerging field of systems immunology. And meanwhile, we have also generalized the NetBait, upgraded to NetBait version 2 to systematically identify hidden driver by reverse engineer network from spark and single cell expression data and for protein activities from multi-omics data then to has been applied to many contexts, some of them which has been published and many more on the way. And we also work with Jiyang and Childr Cloud team deployed Cloud app to the NCI Cancer Genomics Cloud to benefit more researchers. It's actually funded by our Cancer Center supplement. And we have developed many sophisticated visualization and analysis features in NetBait 2 to make a powerful package for comprehensive hidden driver analysis. And we even had a cool logo designed by Yogesh Agada, a talent graduate student in the lab. So NetBait hidden driver inference is at the bulk level. And as we all know, single cell sequence emerged as a powerful technology in dissecting cellular heterogeneity, and we have extended our hidden driver analysis to single cell omics data. Specifically, we have developed a novel pipeline named SC-Miner for intracellular network and hidden driver inference from single cell data. We have been applying various single cell profiling technologies to profile both tumors and enrich tumor marking environment, immune marking environment from patient samples and immune competent mouse models, followed by SC-Miner identify hidden drivers mediate the tumor and tumor immune marking environment. This is an overview of SC-Miner for intracellular network hidden driver inference. And multiple lab members, Adam, Dean, Hoshi, a cohort between Homberg and me, and former member Tracy have contributed and led the development of this pipeline. I won't go into details, basically goes beyond the cluster analysis to infer cluster of cell type specific network and infer the cluster of cell type specific hidden drivers and rewiring events. And the key computational technique we use is mutual information, which capture nonlinear dependence and also Bayesian modeling for robust statistics. Another key feature of SC minor is to infer the cell-cell communication network driven by ligand receptors shown here. And Qin-Chiu Yan, senior bioinformatics research scientist, has led the development of pipeline SC minor to infer ligand receptor-driven intracellular network. For example, as shown here, the ligand expression tumor subclones interacting with the various immune marking environment cell types, macrophage and big cell, express the receptor. Here's one example of application SC minor in hypoglioma in close collaboration with Dr. Jude Baker. We profile a mouse model, K27M driven DIPG model, Jude developed by single-cell RNA sequencing. One of the top drivers identified by SC minor is CFP3601, reported, and validated by a study a couple years ago. If you look at activity, you do see it's OPC like tumor specific. But if you look at expression of this transcription factor, you don't see any pattern at all. That's again the power of SC minor inferred activity to identify the hidden transcription factor drivers. Here's another example in hepatoblastoma, the most common liver cancer in children. It's a wonderful collaboration with Jude. And Childr Pamposa in lab led the computational work in this study. And in this case, we identify a marking environment niche that interacts with tumor cells through chemical ligand receptor and promote liver cancer metastasis. So I won't go into details, but it was published a few weeks ago in case you're interested. Another important emerging technology for our single-cell hidden driver inference is spatial omics, which adds another layer of information, inside information on top of the expression, either RNA or protein. For example, spatial transcriptomics allows to detect the spatial patterns different, tumor marking environment cell types. It shows class, in the single-cell data, but actually many of those cell types inside they don't show those kind of pattern. For example, T cells kind of spread around, but you do see co-localization of different cell types, indicating they might be cells origin or cell-cell communication. For example, IDC-like tumor cells, microglia, macrophage, and OPC-like tumor with the cycling tumor cells. So this is using Jude's DIPG model again, using text genomics with platform. And also computationally also enable us to reconstruct the in situ cell-cell communication networks. And Adam Dean in lab has been developing a spatial SC minor pipeline algorithm for this purpose, and will further filter our ligand receptor-driven cell-cell communication network from a single-cell data. It will be integration of the single-cell RNA-seq with spatial transcriptomics. And I want to give a shout out to a super exciting symposium organized by McDyer, Charlie, Paul Thomas, and myself, and is deeply connected with this Science of Childhood Cancer lecture series sponsored by our Cancer Center, and we have put together a lineup of stunning speakers who are pioneers in the spatial omics field, with expertise in technology, computational algorithms, application in cancer, immunology, and neurosciences. As many of you know that Jude, is building a center for spatial omics as part of the 22 to 27 strategic planning, the task force led by McDyer. And, it's going to be a virtual symposium on June 2nd, Thursday. Register mark your calendar if you are interested. So next, I'm going to share a few exciting translation stories enabled by hidden driver inference from BARC and single-cell omics data. So here are the two stories I mentioned at the beginning of my presentation. Both show that net-based inferred hidden drivers can be translated into therapeutics and can be tested in clinical trials. And the inferred activity can be used as a biomarker to select patient. So I'll briefly walk you through the second story with Jiyang on TL. And that story started with a puzzle Jude had a while ago. Jude had a wonderful pharmacotyping platform to profile drug sensitivity ex vivo using leukemia cells from patient. And one of the drug he profiled is dasatinib, which is mentioned known as ABL inhibitor to treat leukemia with ABL fusion, as shown here. Beautiful response in BCR ABL BL. However, in TL, there's a substantial fraction, especially in pediatric cases, over 44% pediatric TL also respond to dasatinib. However, there's no ABL fusion or translocation in TL. So Jude did all kinds of omics profile, whole genome, whole exome, RNA-seq, even some single cell RNA-seq to identify what's the underlying driver for the second response in TL. Unfortunately, nothing really came out. And it came to us, and Jiyang Liu, a postdoc in the lab, applied nebula algorithm by bringing a TL injectome from the independent target cohort, RNA-seq data about 260 profiles and superimposed to the sensitive resistant profile, RNC profiles. Now we can see the number one nebula infodriver is LCK, and if you look at the possibly enriched by the top nebula drivers, pre-TCR and TCR signaling, that makes it interesting because LCK was reported to be off target of the dasatinib. So that made us to further validate LCK might be the underlying target driver for the second response in TL. Indeed, we validate, by phosphoproteomics with streaming we let our proteomics core as well as phosphoflow inhibition, of LCK and also TCR signaling kinases, up on the same treatment sensitive TL samples, but not resistant ones. And independent of our analysis, Dr. Kim Stackmeyer from Dana-Farber also identified LCK as number one dependency in TL using the cancer dependency map data, comparing with almost 7,000 cancer cell lines, all hematologic cell lines. You probably don't need any statistical to see LCK is outlier, unique to TL. And we collaborated, with Kim. Actually, the collaboration was the pure serendipity. I was presenting one slide, on the second story in Childr SAB meeting a couple of years ago, and Kim was sitting in the room as co-chair and Childr SAB. I did not mention anything about SAK, but 2 weeks later, Kim reached out to Jude, Ash showed this data. And that we did not make it to the manuscript, and we really appreciate Kim's openness, which led to wonderful collaboration. You may have concerns if you just look at the dependence map data because of statistical power, the only 3 TL cell lines. But if you integrate with our analysis, with patient and also pharmacotyping, you have more confidence to, believe LCK might be a true vulnerability in TL. And also, Kim, has led the pediatric cancer dependency map, published the first generation PDAP last year. Super exciting data. And we further validate the dasatinib in vivo using TL patient-derived xenograft model. And indeed, even monotherapy of dasatinib reduced leukemia burden and extend, mouse survival significantly. However, you may notice that almost, all mice will relapse by developing resistance. And remember, there are more than 50% of TL cases we test are resistant, initially to dasatinib. So that goes to the question of biomarker, and clearly you cannot use, ABL fusion or Jiyang has developed a net-based activity-based biomarker, including 30 dasatinib sensitivity drivers to predict the dasatinib response. In our preclinical ex vivo studies, it performs well. Even in the validation cohort, can achieve AUC of 85%. And if you look at the top driver of the sensitivity, we have almost half sensitive, half resistant. And the biomarker is going to be evaluated in the ongoing clinical trials. And if you look at the top drivers, particularly resistant, you notice one of the top drivers is BCL2, which can be targeted by drugs like venetoclax, navitoclax, and other BH3 mimetics. So this might indicate the some resistant cells, might be sensitive to BCL2 inhibitors. And Jiyang happened to profile venetoclax in the TL cohort, and we checked the correlation of the sensitivity with venetoclax sensitivity. Indeed, there's negative significant correlation between the dasatinib activity and also venetoclax activity in TL. And so we can do a negative analysis on venetoclax sensitivity, then check the correlation of the drivers. There's a striking negative correlation between the dasatinib NABI drivers versus venetoclax drivers, and the top ones here, LCK for the sensitivity in BCL2 for venetoclax sensitivity. And almost 8 years ago, Tony Latai, who is expert using drugs like venetoclax or targeting apoptosis pathway, reported that BCL2 has a high activity in ETP, the early T cell progenitor ALL. And also, there's association with the T cell development that motivated us to think, there might be association with the T cell development on dasatinib or venetoclax sensitivity. So we superimpose the T cell development stage signature into our patient RC data and group, by their the sensitivity. You can clearly see that indeed the dasatinib resistant one, the more likely respond to BCL2 inhibitor kind of enriched in the ETP stage, and the sensitive one seems to be enriched in the DN double negative 3 and 4 stages. And we further check the LCK activity as the sensitive biomarker score and also BCL2 activity across here's mouse T cell development stages. You will see that indeed, the ETP stage BCL2 is very high and goes down, significantly. On the other hand, LCK or the certain sensitive biomarker score goes up, beginning very low and goes up, steeply at the double negative 3 and 4 stages. And similarly, in human T cell development, which has a lower resolution, but we still see same pattern in the Simon's 3 and 4 canon corresponding to the DN3, DN40 in the mouse. So that, triggered us that, the association with, T cell development might indicate that, the resistant clones pre-existing in the TL sample that, might explain the, acquired resistance triggered by a single the certain single agent. So, to answer that question, straightforward, we will be doing single cell RNA sequencing, and we did that, with Ben Shan in John Easton's lab, our genomics lab, and how she did all the analysis. And indeed, you see, this is using a TL-PDX model. Indeed, you see there are 3 subpopulations. So we did the SCR single cell RNA without the second treatment. You will notice that the cluster 1 population, cluster 1, after the second treatment, they kind of got enriched, the below one. So indicate they might be the resistant clones. And that way, the cluster 3 kind of got reduced, might be the sensitive one to the dasatinib. Then we can check the LCK activity. Indeed, the LCK activity is strikingly high in the cluster 3, and BCL2 activity is the other way around, high in cluster 1, and there's a striking that correlation between LCK and BCL2 activity. Actually, the 3 cluster kind of corresponding to T cell differentiation, ETP, mostly in cluster 1, then cluster 3 is enriched by all the double negative 3 and 4 stage markers. So that leukemia heterogeneity and also association with T cell development motivated us. Actually, others, got a nice review together with our paper, the idea of targeting developmental origin of cancer. For example, here using BCL2 inhibitor targeted the ETP, early stage clones, and also the second target the DN3, DN4 clones. And also you can naturally think about combination of LCK inhibitor plus BCL2 inhibitor, and indeed validate the second with navitoclax, dual BCL2 XL inhibitor, indeed, combination shows actually additive effects in TL. And more exciting, we have collaborated with Dr. Marina Konoplava, MD Anderson, who is really expert, in using drug like navitoclax, and launched a phase 2 clinical trial to test combination of ponatinib, another LCK inhibitor, with venetoclax in adults with TL. And this story is also a perfect example of hidden driver approach, integrating multiple data, really for translational research. And the data just walk you through is really piece of huge volume data we reported in the paper and also currently generated. And more exciting, multiple clinical trials across the world are being launched or has been launched. And I sent Jude. MD Anderson mentioned Europe and China. Many of them are coordinated by Dr. Pui. And I want to give a shout out to June's talk in this series on May 5th, when I believe he will show you some exciting fresh data on patient response from some of the ongoing clinical trials just mentioned. So in the next few minutes, I'm going to share with you another exciting story in pediatric solid tumor, specifically rhabdomyosarcoma, the most common soft tissue sarcoma in children. And it's a wonderful collaboration with McDyer and also the solid tumor program. Rhabdomyosarcoma, based on its pathology or genomic profile, can be classified into two subtypes. Alveolar rhabdomyosarcoma, featured by PLEX-FOXO1 fusion, and also embryonal rhabdomyosarcoma or fusion-negative sarcoma. So these two subtypes behave very different in the clinic, and there's urgent need to for novel therapies tailored by the subtypes of rhabdomyosarcoma. So one approach, clearly we had is a hidden driver approach. So Xin Huang, a postdoc in the lab, has worked very closely with Anand Patel, who is extremely talented MD-PT, a molecular oncologist, and Xiang Chen in our department, and integrated the single nucleus RNA-seq data, non-profiled, of about 12 embryonal rhabdomyosarcoma and 6 alveolar rhabdomyosarcoma patient samples. And with the bulk omics data, RNA-seq, proteomics, phosphoproteomics from the our PCGP and PCGP2 project, Max team has profile, but integrate them together to identify either ARMS or ERMS specific hidden drivers. Before showing you the hidden driver, just mention that rhabdomyosarcoma is a tumor that forms on skeletal muscle, which follows the myogenesis development from the stem-like mesoderm population to myoblast to the differentiating myocytes and myotubes. So one question we had, similar to the TL study or inspired by the TL study, was that, is there any intratumor heterogeneity? And also, if the intratumor heterogeneity is correlated with the development myogenesis here? And the answer is yes, based on the single nucleus RNA data analysis. And interestingly, there's a surprising difference between embryonal versus alveolar rhabdomyosarcoma, in which that the mesoderm-like population, tumor cells only exist in embryonal rhabdomyosarcoma, but not in alveolar rhabdomyosarcoma. So that makes interesting, and also you can see those kind of cell proportion only exist in the embryonal rhabdomyosarcoma. So now going to the hidden driver inferred by Jiyang using the bark RNA-seq data. One of the top drivers was EGFR. It's a tyrosine kinase receptor, multiple approved drugs, to treat EGFR mutant lung cancer and many others. And more interestingly, if you look at the single cell data, particularly EGFR activity across different, cell stages, is uniquely high in the metaderm, which is unique to embryonal sarcoma as well. And if you look at the HD map, it's HD map is algorithm developed by Adam in the lab to utilize the network topology, and minimal spanning tree algorithm to understand the driver, outer driver. If you look at the map, HD map, specific to metaderm, you will see EGFR is one of the very, very important hub, indicating that EGFR might be a therapeutic viability target in metaderm like embryonal sarcoma tumor cells. And just want to mention that it's not driven by EGFR mutation, like those tumors that use EGFR inhibitor. And EGFR we can easily test by EGFR inhibitors. There are multiple options, and a tested using two EGFR inhibitor. If you just use EGFR, this is a 3D model of embryonal sarcoma. If you just look at the EGFR inhibitor alone, it does not do much because it's only activating the metaderm population, which is kind of rare in the whole bark tumor. So you have to combine with another drug that eliminate, the myoblast, myocyte, tumors, and low-hanging fruit actually is chemotherapy. So it's a combination of EGFR inhibitor with chemotherapy. Now you can say the actually it's additive effects again to slow down the tumor growth, and we validate that in using topic xenograft model as well. So other preclinical validations being conducted by a Max team. And also other therapeutics that target the myoblast population, for better combination are being developed. Hopefully, can be translated into potential clinical trials for rhabdoid sarcoma. So the data I just walk you through is really just tiny piece of huge, comprehensive single cell and integrated analysis of Xiang and Xin has put together, and it was just accepted. If you're interested. If you look at put the two stories together, one on TL, one on rhabdoid sarcoma. Interestingly, they share a common theme. Number one, they both have intra-tumor heterogeneity, no surprise. Number two, the intra-tumor heterogeneity actually associated with development. This could lead to a combination therapy, strategy. We name it as clonal therapy, which targets multiple tumor subclones driven, likely by developmental stages, as I showed you in the two examples. And this along with the development of combination therapy that could really lead to curative cancer treatment. But beyond the two stories, there are a couple other questions we ask ourselves. The two stories just showed you, they both started from a single driver. But we ask ourselves, can we start, predicting pair of drivers and starting the combination at the very beginning to minimize the resistant relapse? And number two, both stories, focused on tumor cells, but we all know in real patient tumor is never isolated. There's tumor marking environment. That's why, immunotherapy has proven to be, if not the most promising, one of the most promising cancer treatment strategies, because of the ability to induce durable response, although it only works in a small footprint patient. So not ideas to identify drugs target tumor immune marking reactions. So to address this challenging questions, I'm going to present a few ongoing efforts to systematically predict paired, of synergistic driver and corresponding drug combinations. We have developed algorithm named Simba, spearheaded by Jing Liu, with collaboration with Jiyang, something in the lab. So our use group stream medulloblastoma, wonderful collaboration with Dr. Martin Russell and with tremendous support from Dr. Maggie Jar as example to explain this algorithm. So using that bit, we can easily identify, group 3 medulloblastoma specific, drivers, and we can come out, a short list. For example, here 86, we call seed drivers. Then for each seed driver, we can survey, all the driver candidates and search for partner drivers, B, whose subnetwork is interacting with the seed driver subnetwork, and combine the two networks, explain the, phenotype better statistically. And then we can derive a driver combination list. So by mapping to a curated drug targeting database, we can transform the driver combination list into a drug combination, short list. And for brain tumor specifically, we collaborate with Laura Rankwitz from CBT to curate a brain penetration, database, because blood-brain barrier in brain tumors that you can to further narrow down the compound list. So given the short list of compound pairs, then we can design a focused drug, small drug screening, followed by in vivo validation. Instead of random sequence compounds, then we do a focused approach. So we did that. We did a first screening only with 320 drug pairs, including actually only 20 seed drugs and 16 partner drugs, with 2 doses per drug. It's kind of first raw quick screening. And then we focus on the top 10%. We scored high, 32 pairs, with a deeper screening with 10 doses per drug. Now out of the 32 pairs we screen, surprisingly almost 60% pair, 32 pairs showed synergistic effects statistically. And there's outlier combination that can be actually it's being followed by in vivo validations as well as in more cell and also in vivo models. So the Simba can serve as a way to derive or predict, potential combination pairs in a specific context, for example, group 3 medulloblastoma. Now you can design a focused library, so that will, dramatically improve the screening rate, as I just showed you here. So for the development of combination with immunotherapy, and there's a clear unmet medical need, especially for pediatric cancer. Despite the remarkable success in adult cancer, the immune checkpoint blockade is effective only in about 20-30% adult patients. Especially, unfortunately, shows no efficacy in pediatric cancer so far. Part of the reason actually is because anti-tumor immunity is not just T cells or PD-1, PD-L1, CTLA-4. Now LEX-3 was just approved. But there's an increasing, appreciation of the complexity of tumor immune marking environment. So it's no longer meaningful just focus on tumor cells or just focus on one subset of tumor marking environment. You have to look at them together. And moreover, the CAR T therapy, which shows, effective in BL, unfortunately shows limited efficacy against pediatric solid or brain tumors. And partially due to the highly immunosuppressive marking environments. And there's a great need to, identify exposed hidden driver that can improve, CAR T infiltration, persistence, and resistance, so then you can improve the CAR T efficacy against solid and brain tumors. And one critical step and challenge is to define the tumor and tumor marking environment interaction map in pediatric cancer, especially in solid and brain tumors. So to address this challenge, together with Hombo, Dr. Terry Geiger, Stephen Gottschalk, we have put together a blue sky proposal named iTargets. The idea is to apply 2 independent but highly synergistic strategies, including the the one I just described you, the hidden driver approach from BARC and single cell as well as spatial omics data. And the other one is the super powerful functional genomics, in vivo screening. Many of you probably have heard from Humble 2 weeks ago, followed by preclinical modeling and validation translation studies pioneered by Dr. Geiger, Dr. Gottschalk. And the goal is really to build a high-resolution, reference map of tumor microenvironment interaction in pediatric cancer, and identify the underlying hidden drivers, and also to enable and advanced curative immunotherapy for pediatric cancer. And we are extremely grateful to Childr leadership for the approval of this project. It's been a year and a half. And of course, we're not going to work alone. And we have been aiming to establish an in-depth ecosystem, at St. Jude, a collaborative ecosystem by working with, colleagues across different programs, cancer center, departments, and course to facilitate, fundamental discovery and translation studies for oncology, pediatric cancer. And we have made exciting progress, for iTarget project in the past year and a half. And hopefully we'll update you soon in the near future. To summarize, I hope I convinced you a little bit that the hidden drawer approach I described you today could be useful to identify, drivers targets that conventional genomics or, a different expression-based approach may fail to capture. And, infer hidden driver and also activity from expression data can become, hopefully the next generation targets that goes beyond, genomic alteration biomarkers, to select or stratify patients. And the combination therapy is targeting, hidden drivers in different tumor clones, associated with different developmental stages, all targeting tumor immune microenvironment interactions, will likely be powerful, therapeutic strategies to really minimize drug resistance and relapse and metastasis, for cancer treatment. And lastly, as system biologists, we're not only reverse engineer biological networks, but also building reliable collaboration networks. I'm extremely grateful to all the support, Baker Jinghui, who hired me and brought me to St. Jude. All the wonderful collaboration with amazing, collaborators at St. Jude and outside. And the stories that present actually just a few among many exciting projects. I have been extremely fortunate to recruit and build a team with talents from diverse backgrounds. Also grateful to all the funding, both internally and externally. And also we're extremely grateful to patients and their families who have trusted us and donated their precious biopsies for our research. It truly takes a village to move things forward, especially for translational studies. And Dr. Imar Gijar, who is my clinical manager in pediatric brain tumor and also our chief neuro-oncologist, often told me that when PIs don't get along, patients die. So we can always find ways to work together because we share the same mission, finding cures, saving children. Well, if you look at the outside world, when countries don't get along, people die, innocent people die. Well, eventually peace will come back, as history has taught us, because most of us share the same belief, in peace, in love, and hope, come back sooner for those who are suffering in the war and, finding peace, living people. And, as the pandemic is coming to an end, finally, hopefully we'll have more in-person activities, like the one we had before the Delta surge, and more in-person presentation, which we have missed a lot. With that, thank you so much for your attention. I'll be more than happy to take any questions.
Thanks, Jiyang, for the great presentation and really shows the power of a hidden driver-based approach on multiple cancer types as well as in basic science related to immunology. So there are a few questions coming through. The first is from Marion Evans. It is related to the rhabdomyosarcoma sarcoma study. Does the underlying mutation influence the effect of EGFR inhibition? Does this work across multiple PDX or cell line models?
Good questions. For the mutation, I'm not sure enough, Xiang has we have looked into details, but it could be potential reason that drives EGFR activation. But at least, it's clearly not EGFR mutation. And yes, I only show you one, actually two models, one in organoid, one in PDX. I believe Anna and Max team has been testing in multiple embryonic rhabdomyosarcoma models.
Great. The second question is from Shang Chen. It's also related to the rhabdomyosarcoma story and the EGFR. Great talk as usual. What do you think we can do to identify potential targets for the alveolar subtype, which has few mutations but a poorer prognosis?
No, I mean that's a puzzle to me, probably to the whole solid tumor program as well. So my thoughts are the following. So in clinic, actually the elevator does worse than embryonal. I think the two potential reasons. One is the myoblast population. I didn't know if you zoom in on myoblast, there are subsets, the Western one and also the second one. So I think either one this is a liability for bad worse prognosis. The other is the market environment. I did not show any market environment data. He has been working on that extensively. We did see striking difference in terms of market environment landscape from the single cell data. That's another potential reason that could explain the prognosis difference between elevator, particularly in elevator we want to identify more potential novel either target or combination with immunotherapy. So yeah, I think together we are working on that.
Yeah, I also have a question for you, Jiyang. So the LCK analysis is really exciting and fascinating, and as you have shown, the ETP status, right, appear to be related to the efficacy of LCK. So I'm just wondering if you can comment on in the clinical setting how we can run NetBid to identify the patients at the diagnosis, not only at the relapse, to find these that will be amenable for LCK analysis, or in general, how the network-based approach can be implemented in the clinical setting.
Yeah, thank you, Jinhui, for the question, for the sharing the thoughts. Yeah, exactly. So far, as you are leading the cleansing, team, so I think we should, go beyond just look at mutations or fusion from the RNA-seq data. I think the activity, inferred by our, network and that approach can, could be a potential powerful, biomarker to really, predict the response, when the patient, because we're doing the RNA-seq, in a clean, environment, so I think if we can, but there would definitely a lot, development to make sure it's a robust pipeline, get a robust, predict, at least, scoring from the RNA-seq data. So I think we probably have to work together, with you, cleansing and also pathology and also, Dr. Bowie, the whole, Charles, to really think how, that would be really the leading, efforts. No one has tried yet, but I think preclinically we already demonstrate the activity, not just expression, can be a powerful biomarker to basically predict response that could benefit a lot of more patients that does not have able mutation or others. So I think that would be a kind of my kind of dream, if possible. So thank you.
Yeah, so another related question is about how you control false discovery in the NetBid analysis. Can you, because I know there's a lot of the phenomenal findings you reported, but I just want to hear a little bit on the technical side. What's your thought on this?
No, absolutely. From a statistical point of view, the false discoveries, will always mean so. So basically, in every step, we always have the false discovery, calculation. But we also be mindful that false discovery many times could be misleading. So many of the hidden drivers we identify. If you look at false discovery, it might not be that encouraging, but the validation data, definitely shows they are important. So that, goes to the question, how much, you should leverage, statistics and also the real biology. So, that's really depend on, it's a multi, task correction. The number of tests, the number of drugs you test matter, right? And there are others, the distribution also matters as well. So we have a couple strategies, but we use FDR as a reference, but in many cases, we might go to the raw p-value and later, to check the FDR. At least to make sure, it's reasonable, not too just random something. But it's also a progress, work in progress, how we can really develop statistically sound procedure to infer the activity and also the cloud inference. But it's also in work in progress. But thanks for the question. Happy to discuss if you want to talk details or have better ideas.
I also just want to share a comment from Corinne Feist. Thank you for the fascinating study and the presentation today, and she's looking forward to attending future programs.
Thank you.
If there's no other questions, I just want to mention that, we're going to thank Dr. Yu for this excellent talk, and we will now close this event. But I just want to mention that next week, March 31st, we will have Dr. Katie Jingwei from Dana-Farber, Boston Children's Hospital, to present learning from the children designing better clinical trial with their data. So at this point, I would like to thank Dr. Yu for the great talk and all the participation of the audience in this very engaging and exciting discussion. So thank you all, and I think we can close a little bit early today.
Thank you so much. Thank you, Jingwei. Really appreciate that. Thanks all.